Hi, hello, and thank you for clicking on my video. I'd like to share a little bit about my research, which uses whole brain modeling to understand deep brain stimulation. So let me start sharing the presentation. All right, I'm Jill, and I'm a postdoc in the brain simulation section in Berlin at the Charité and Berlin Institute of Health under the guidance of Professor Petra Ritter. And we use the virtual brain. It's a specific software internationally used for whole brain simulation. And to make it even more complex in this first talk of this session, I'd like to introduce not only whole brain modeling, but combine it with spiking network modeling in a so-called multiscale model. But first, let's see what it is about. I have no disclosures. And the virtual brain. It's basically a network. As we know, the brain network is its essence. It's the input to the virtual brain, the structural brain network, also called connectome. And then every brain region is a dot here, and the connections are links. And on every dot, we place dynamics. And how we place this dynamics is in its average simple form. It's like this bird swarm. We just trace the average activity of neurons, and the average is called in physics mean field theory, and the mean field model has been developed for a multiple number of neurons. There's an example. So each region will have, for example, this is a DECO mean field model, an excitatory and an inhibitory population, and we will describe their interactions on average. And then we will connect those different regions, and we will see what complex dynamics emerge out of this networked mean field models. So here you can see that the mean field model is not magic, but it's often just a number of 5 or 6 differential equations that describe different state variables over time. Those state variables may be, for example, the firing rate of each region. And then when we connect it with a so-called global coupling, we will see that each region influences the other region, and together they will end up in different dynamic regimes. So what comes out of the virtual brain when we input these equations and the mean field model is what you choose it to record. It can record local field potential activity, so raw neural time series, or it has also embedded monitors of MEG or EEG data, or even bold fMRI. And the most prominent example currently of the use of the virtual brain in the clinic is the Epinov trial from France, where they started using a virtual epileptic patient, short VEP, to study before the surgery epileptic patients and see what will happen once they get this area that is resected in the surgery removed, and they try to predict if that will make them seizure-free or not. So those are patients being medication refractory, so they've tried everything else before such an invasive surgery. And you can see that the normal procedure gets a false discovery rate of about 0.5, so meaning that patients will be non-seizure, or that patients will be non-seizure-free in some cases and seizure-free in others. So here, the green one is the seizure-free patients, and there we have a false discovery rate of about 0.3 for the currently used method. And you can see there is an outlier, but in average, the virtual epileptic patient has nearly a false discovery rate of 0% for the seizure-free patients. So these are preliminary results of this first trial with retrospective data. Now it's running prospectively with over 400 patients and trying to help and guide the neurosurgeons in which area should be removed to get patients seizure-free. So that's about it for the virtual brain. And now let's make it even one step further and more complex by introducing a multiscale model, meaning that we still have the virtual brain up here. We have the brain network with the mean field models for all cortical areas. But now for our special use case of deep brain stimulation, we needed a spiking network model as well that describes each neuron's spike trains in very much detail. And we chose to model this zoomed in version of the virtual brain with Anarchy, which is a modeling software developed by our collaborators in Chemnitz. and it can model, just like NEST or Neuron O'Brien, every single neuron's activity. For these regions, which are in the basal ganglia region and the thalamus region, we chose to make it more detailed. And we can have, as an output of this region, single dots, or here single spikes of single neurons. Whereas output of the virtual brain for the cortical regions are average activity or average firing rates. Now, what can we do? We can generate spikes in the one direction to make the virtual brain talk to the spiking simulator, and in the other direction, we just count the spikes to get our usual average firing rates. And this is how these two simulators communicate and translate among each other. This is the extension of the virtual brain called TVB multiscale. And we've recently published this first use case together with our collaborators, now focusing on deep brain stimulation. So what is deep brain stimulation? It's an electrode that is inserted deep inside the subcortical structures of the brain, often the subthalamic nucleus or the globus pallidus internus. And this is approved for many different neurological diseases already, and is helping patients to reduce, for example, tremor symptoms in Parkinson patients. So by emitting some electrical pulse to the surrounding tissue, it can help patients. But nobody understands yet completely how this mechanism works and what are its effects of this small-scale mechanism, this very local mechanism, on the global scale of brain dynamics. Some patients get language slurring. So where does it come from? How does this small perturbation of the network spread and extend to the whole brain network? This is exactly what we aim to target as a future research question. In this first proof of concept study mentioned below here, we have seen multiple effects on the different scales when we switched on our model for one Parkinson disease patient, also had a healthy control. So we saw that the Parkinson disease patient, for example, had the thalamus firing rate quite low compared to the control. But once we switched on the virtual deep brain stimulation, so we gave a stimulus to the STN or the GPI, we saw that it was normalizing back to the healthy level. So we could disinhibit the thalamus, just like it's known what's happening from experiments on animals, mostly from literature. But then the cool thing was that also at the same time, we can now see what's happening in the rest of the brain, and we can monitor the activities of motor and frontal regions, as you can see here. And we have found quite some effects. For example, the postcentral gyrus, which is normally less active in patients than in controls, is getting reactivated when we switch on the electrode, turning red or even black for different stimuli. So all of our code is public. The data for that paper is also public, and feel free to have a look and get in touch with us if you want to do something with this model. But so far, this was a comparison of firing rates with literature. What we then did is now extend the model, and these are preliminary results, towards extracting bold data and a bold response of what we call the virtual DBS model. And when we simulate and when we then compare DBS on minus DBS off activity, we can see different changes in the bold activity of different regions here. These are the regions of the basal ganglia and thalamus again, and these are the cortical regions. So now we got ourselves bold data to compare it to empirical from patients being scanned on and off. Unfortunately, this is just n equals 2 so far. It's very much ongoing work. And you can see the common changes in red when activity increased in those two patients, and blue when activity decreased in those two patients. You can see that, for example, for the occipital lobe, we do already quite well, and our simulation is predicting what's happening in those two patients. But we also see that our simulation actually seems to be much less scattered and much more equally spatially distributed in its effects, while the real-world patients here are only 2, but seem to have much more individual effects. So when we saw these results, we knew we needed to move as the next step towards the individual modeling, going away from averaging over multiple patients and having a group average virtual DBS brain. However, when we compare functional connectivity, so this is single region activity, And this is now, as we know it from these functional connectivity matrices, an entry describes the communication between the two areas. Then we looked at those two patients, and we looked at whether their communication increases or decreases due to DBS in both of them. And we boxed in every change that's happening in both of them in the same direction. So you can see there are 5 boxes in green and 1 in red. This means that the green boxes, we were able actually to capture exactly this change that's happening in both patients already with our simulations. The red box means that we predict a decrease in activity of the communication between thalamus and S1 here, for example, but the 2 patients had an increase. So there is some fine-tuning of our model to do. And just to show you how it looks currently in our model, we have this biphasic stimulus, which is emitting this to the surrounding tissue. And you can see that it's happening a lot of changes in the beginning in the basal ganglia regions, but you can see decreases and also now some increases coming up in motor and even frontal regions with a certain delay. So we see this effect of the stimulus in our whole brain modeling, which is actually quite cool, thinking that it's a tiny stimulus to just these 200 neurons that we model in the STM. All right, and then the ultimate goal is, of course, to link whole brain modeling to behavior. So how did we attempt this in this preliminary research results so far? So we have now the individual location of the electrode, and we developed an approach where this individual location can be taken as an input to the whole brain modeling. So then we simulated with this individual location, activating individual fibers, which then lead to regions being stimulated. And we simulated this for these 18 patients. And then we looked at their principal component of their dynamics, of these simulated dynamics. And as you can see, the principal component analysis shows that those patients place on different regions within the principal components, which is good. So we see diversity, we see an influence of the electrodes contact that's active and our whole brain modeling, which is again great to see this effect. And at the same time, we had data of the real patients being stimulated with a certain amplitude and a certain contact, and then being measured concerning their motor skills on a so-called rotor-meter task. So we knew what is their clinical improvement and what's their, if they are better doing clinically when they have the stimulation setting or doing worse. When they're doing worse, it's blue, and we can see that all the blue dots are down here, which is great that our simulation kind of also puts them in the periphery. And then we can also see multiple measurements of the same patient, which are connected with an error. And we calculated the average error over all these errors, meaning in this direction, which is the upper left corner in our case, we see that patients are doing better clinically. So it's great that we have a first link, that our simulation is somehow identifying a preliminary region here, where when we see those dynamics, we can link them to clinical improvement. What are the smileys, you might ask yourself? The smileys are the clinical settings, optimal clinical settings that the patient has on a daily basis. So we cannot yet find a certain area for those. And we were wondering a lot about this, and this is really preliminary. But we can see that they are in between the dark red ones and the blue ones. So it seems to be an in-between region of this clinical motor improvement task, which is, of course, a very specific task. Where the patients, when they have their daily settings, do okay on this task, probably. And there is an even better setting for the specific task, but that might not be ideal for other daily activities. So this is a current preliminary interpretation. We are very much on this, getting more and more data every week by patients being scanned here, working closely, collaborating with the clinicians here. All right, so we already identified an interesting principal component, which is this principal component 3, which shows the activation and which shows then when we see this pattern in our simulation, it seems to be related to doing better clinically. And that's the first step towards using these simulations to test in silico different electrode settings before then testing them on the patient. And now a little preview that we are also working on TMS, not as advanced as the organizers of this workshop, but we started with it, and we are also excited to combine it in the future with the DBS research. And you can see here a simulation of resting state, and we will certainly see such a video later on as well. And with TMS, it's non-invasive. So it's starting a TMS pulse now. And what we can already see from our simulations here is that you see those waves, you see these evoked potentials that we are used to of excitation and inhibition after a TMS pulse. All right, and with that, I'd like to thank my collaborators. I've already mentioned the large group of clinicians that's helping us to get data for this, and of course, my colleagues from Berlin. And at the same time, if you'd like to perform virtual DBS yourself, we have, as I said, everything online and also published YouTube tutorials to get you started. It would be great to get in touch and hopefully see you soon in Montreal.